Good morning, everyone. Good morning, everyone. A hearty
Good
welcome
morning.
to you all for today's session on another session on QbD, concept of QbD, which is organized by RBVRR Women's College of Pharmacy. So I, Dr. KV Ratnamala, on behalf of management, faculty, and students of RBVRR Women's College of Pharmacy, welcome you all for today's third session on application of QbD principles to biopharmaceuticals, formulation and development by Dr. G. Girija Shankar Garu. It gives me a great pleasure in introducing our today's speaker, my guru, Professor G. Girija Shankar Garu. Sir has completed his B.Pharm, M.Pharm, and PhD from Andhra University. Sir is a merit student from start of his career itself. He has got SSC Merit Scholarship from Ministry of Human Resources and Development. He stood fifth in BPharm and he was awarded J.J. Rao Best Thesis Award in 2000, Dr. Sarvepalli Radhakrishnan Best Academician Award in 2017, AP State Best Teacher Award in 2019. Sir is having a total teaching experience of 27 years. He started his career as a teacher in Vishnu College of Pharmacy in the year 2003 as an assistant professor. Later, he was promoted to professor in pharmaceutical technology in same college. Presently, he is working as a professor in pharmaceutical biotechnology division, Andhra University, Visakhapatnam. Sir is also having an industrial experience. He has guided around 83 MPharm students, 21 PhD students, and he is having almost 2 DBT projects to his credit and 1 ASCT project to his credit. Sir has guided around 83 MPharm students, and he is having around total 70 international publications and 7 national publications. I welcome you, sir, for delivering today's talk on applications of QbD principles to biopharmaceuticals, formulation and development. I heartily welcome Dr. G. Girija Shankar Garu, Chairman, Faculty of Pharmaceutical Sciences, Andhra University, to deliver his talk today.
Hello, am I audible?
Yes, sir, you are audible, sir. Sir, I think connection got lost, sir.
Yeah.
Yeah, now it's audible,
Hello?
sir. Now it's audible. Yes, sir, you're audible, sir.
Audible, yeah. Right. Thank you, Dr. Ratnamala, for introducing me. Right. So, first of all, let me thanks to your college, RBVR college management and the principal, Dr. Sumakant, for giving me this opportunity to deliver a talk on application of quality by design principles to biopharmaceuticals, formulation and process development. Right. So good morning to all of you. So this is a talk which I am going to give you today in my session, that is right. I think all having audible, everything is okay right now.
Yes, sir.
Right?
You're audible, sir.
Right.
You're audible,
Right.
sir.
Right. My screen is also visible.
Yes, sir.
Right.
Your screen is
Right.
also visible.
Right. So let me first say about that is what is this topic, that is application of quality by design principles to biopharmaceuticals, formulation and process development. So in this topic, that is quality by, there are two terms mainly which we are going to come across in this whatever this one is, quality by design and biopharmaceuticals. So these two are very, very important in this one, and from this we are going to start our this one. Actually, I'm going to give my contents of the presentation, which we are going to say, that is, you see, these are the contents which I am going to today's in my session, which I am going to present. That is, one is, that is first definitions, the terms which I am going to use in my whatever the topic, and then what are the quality characteristics, right, of a pharmaceutical product, and then what is the quality by design, which you have already known But once again, we are going to refresh here what is the quality by design, and then elements in quality by design for product development, that is. So here we are mainly concentrating on formulation development and process development. Both are going to be involved, that is, for a biopharmaceutical. So what is biopharmaceutical development? The next one is what we are going to say. And then manufacturing process of a biopharmaceutical, then formulation design space development, that is, and then risk assessment and risk levels, then process characterization and design space development, and what are the advantages of this quality by design, that is. So this is my contents which we are going to go through in today's my session, that is, right. So first of all, we will just go through that what are the terms which I am going to use in this term. Is the first and foremost one is, because we all know that there is a name, that is pharmaceutical product, that is the regulatory term, that is what we are going to call it as pharmaceutical, for whatever the product which we are going to call it as, that is a dosage form, that is. So here we are using another term, that is what we are going to call it as a biopharmaceutical. Yes, it is also a dosage form, but what is a biopharmaceutical? Because you should have a clarity. In olden days, we are having that name is going to be given as a biologic, that is biologic preparations, that is. Biologics means that whatever the dosage form or whatever the pharmaceutical products which are coming from the biological origin, that is. So in olden days, we are going to you prepare these biological products from the animal tissues, for animals as a source, that is, or whatever the organs or maybe the tissues, we are going to extract it from that. We are going to obtain whatever this product which we are going to call it as a biological product, or any type of microbiological products which we are going to obtain from the microbiological source. That is also what we are going to vaccine preparations in olden days, right? And in the similar way, the other type of whatever the substances which we are going to obtain from another human being, that is what we are going to call it as a blood and blood related products. So these are all comes under biological products, but what we are going to obtain from this is from a biological source, that is. But now the term has been extended, the definition has been given in another way, that is the biologics, which we are going to call it as a biopharmaceutical, that is. So what are these biopharmaceuticals? So biopharmaceuticals are the substances obtained from the biological sources to treat the diseases manufactured by the latest technologies which we are going to use in 19, I think from 80s onwards. That is, actually it was established after the whatever this technology is from 1970 onwards, but in 1980 to 90, these technologies are going to be used for the preparation of these biopharmaceuticals, which we are going to call rDNA technology or Hybridoma technique, that is. So I think I will just give you a brief note on this recombinant DNA technology or Hybridoma technique, because these are now very, very important when we are going for QbD for biopharmaceuticals. Recombinant DNA technology in olden days, when we are going for animals, using that whatever the biological source from the animals, it is we are going to obtain that whatever the required quantity of whatever this biological preparation is going to be very, very less, because we have to sacrifice so many number of animals. We have to take whatever that organ or the tissue and mince it and extract it, and then we are going to obtain the quantity of whatever the biological substance, which is very, very less quantity. And the another important one is the purity is also less. So in such conditions, it is not at all possible to get a highly purified biological product. So once this recombinant DNA technology, that means using a living cell, these substances are going to be producing. How? By this combining the gene of interest, whatever the gene which is going to be having the capability of producing this whatever the substance, it may be a hormone or a protein or any type of blood related products, right? So that gene is going to be inserted into another gene, that means of whatever the host that is. So that is recombining, that is what we are going to call it as a recombinant. So this recombinant we are introducing into the host. That host is, it may be a prokaryotic cell or a eukaryotic cell. So once we are going to introduce into the host, now the host what we are going to containing this whatever the recombinant, which we are going to call it as a genetically modified organism, what we are going to call it as a GMO, that is. So this may be a plant cell or an animal cell, which we are going to use this recombinant DNA technology for production of whatever the useful substances which we can treat the diseases in a large quantity and in a highly purified form. That is the advantage with this recombinant DNA technology. And another technique is, nowadays we are going to have this hybridoma technique. So I will just give you the brief on this. What is a hybridoma technique? That is actually the cells in our body having that whatever the capability of, due to this whatever the immune mechanisms, now the cells are having a capability of producing a substances which we are going to call it as an antibodies, which can have the capability to fight against the antigens. So what these antibodies are going to be, that is these antibodies will destroy that whatever the antigens, it may be microorganism or any type of virus or whatever it may be, that is, right? But sometimes what is going to be happening, these antibodies production also leads to sometimes a complication, enormous growth, because these antibodies are not at all produced at a right time, that is. So sometimes these are going to be produced in an extraordinary conditions, that is. So that means cell is in an abnormal condition, what we are going to call as cell proliferation, what we are going to call as a cancerous tissues, cancerous cells, right? Tumor cells. Now these tumor cells are going to be having producing these type of proteins where this growth is going to be increasing very high rate, at a very high rate, that is. So that leads to carcinoma, or what we are going to call the cancer, that is. So in such conditions, we have to stop that whatever the activity of this abnormal cell, that is, or we are going to call it as a transformed cell, that is. So for that purpose, the whatever that antibody actually produced by the whatever the normal cell, which is not sufficient. So in such conditions, what we have to do, we have to take that antibody from outside. So how we are going to produce this antibody means, that is by using this hybridoma technique, that is. That means actually normal cell is not having a capability of producing this high amount of whatever the this antibody, but there is another cell which we are going to have, which cannot have the capability of producing this antibody, but which can multiply enormously. So these two cells we are going to take. One is normal cell, another one is whatever the cancer cell. These two cells are having two specific characteristics. Normal cell is having a characteristic of producing that specific antibody, whereas this whatever the cancer cell is having a capability of producing, multiplying very fastly. That means this is having one type of gene and this is having another type of gene. So now what the scientists have done is, they are just combined these two cells, that means fuse these two cells, that is whatever the normal cell and whatever the cancer cell. So once these two cells are fused together, then it is going to form that whatever the hybrid cell, that is. So now this hybrid cell contains the gene of having producing the antibody and the gene which is having a capability of multiplying very at a high rate. So that what will happen, it is going to call it as a hybrid cell. So that once this hybrid cell is there, so then this hybrid cell is going to be by in vitro cell culture conditions and by whatever the fermentation process we will discuss afterwards, so that we will grow these cells enormous quantity, and then these cells are having expressing that particular antibody, whatever that protein. Now this protein is now we are going to use for treating the diseases. This is what we are going to call it as a hybridoma technology. Nowadays, whatever these antibodies which we are going to produce specifically from that cell, that's what it is going to call it as monoclonal antibodies. So these monoclonal antibodies are producing by this hybridoma technique, that is right. So now you understood that is it may be a monoclonal antibody or recombinant DNA products which we are going to manufacture, that is from this biological source. That is what we are going to call it as a biopharmaceutical. Now understand, I think this is now this hybridoma technique is now getting a very expanded, that is a research technique, research tool, where we are going to prepare this monoclonal antibodies. In our India, we are having a very less facility for manufacturing of these monoclonal antibodies, except very few companies, 1 or 2 companies, Biocon or some of them are going to be manufacturing. But till today, in this corona's pandemic situation, we are exporting that tocilizumab, which is used in case of corona severe condition patients, they are using. But totally this is export oriented drug, that is. We are not getting in a sufficient quantity to meet our requirements in the present situation, that is. So these technologies we are going to use for this recombinant DNA technology and the hybridoma technology, where we are going to use for the preparation of these drug products, which we are going to call them as a biopharmaceuticals. Now you have to clear-cut concept about what is a biopharmaceutical, that is. So these biopharmaceuticals manufacturing, that is in the development, and in case of whatever the process, we are going to use this quality by design concept. So what is this quality by design concept, that is what is meant by quality by design? Because you are already come across this whatever the definition, because the previous days also they have done a lot of topics are going to be taken by various speakers by this, but I will just quickly go through this. What is this quality by design, that is? So this quality by design is a systematic approach to development that begins with a predefined objectives. That is very important. We are going to develop that whatever the biopharmaceutical with a predefined objectives, and we are emphasized mostly on product and process understanding and process control based on the sound science and risk management, that is quality risk management. So that means we are just going back afterwards, that is this is what we are going to call it as a quality by design, right? But before we are going to start this quality by design, you should have a clarity about what a quality is. That is very important. Quality means, that is, it is, what is the dictionary meaning of quality means? Quality means we are going to say that is a high level excellence. Or when we are comparing with any other product, yes, the product of this is a good quality. So that means in our whatever the pharmaceutical product, what a quality means, that is we are having a very 3 important characteristics with related to the quality of a pharmaceutical product. Anyhow, we are going to discuss. That is one is a therapeutic efficacy, and second one is safety, and third one is stability of the product. These 3 are very important one, that is. So for this purpose, now the quality concept came into existence in pharmaceutical products, that is. So actually previously, that means I hope before 1980s, we are having a concept of quality control only. Quality control means what we are doing, once we have manufactured the product, then we are just check whether it is meeting the specific standards or not of the finished product. That's all. So that means it is quality control is nothing but what? It is end product testing. End product testing, that is. So that is means if anything goes wrong during the process or during the preparation, we are not knowing about that one. So only we are testing at the end product whether end product is going to be passing as per specifications or not. If it fails, then you have to throw it away. So what is we have to improve the quality in what way means? That means we have to give assurance to the customer as well as to the industry, manufacturers, whoever that is, keeping in the customer to reach to the customer, or maybe in the retailer, we have to always give that, we have to meet that whatever the quality of the product. That means we have to assure the quality. After that, in 1980s, this concept has came into existence, that is quality assurance. That is, we are assuring the quality of the product. So in that, we are not testing the end product. What we are doing, we are inbuilding the quality into the product. We are inbuilt the quality into the product. That is. So how we are going to inbuilt the quality into the product means at each and every step, we are going to take utmost precautions. That means whatever the measures, preventive measures, not to take any type of errors or mistakes, what we are going to perform, even procuring of raw material onwards till it reaches to the consumer or to the customer or to the patient, what we are going to call this. So this concept came into in 1990s afterwards. That is what we are going to call it as nowadays. It is going to call it as total quality management, TQM. That is. So this total quality management now is taking a utmost important place in whatever this manufacturing of whatever our pharmaceutical products. That is. So that's why now what is going to be involving it is the total quality management. We are already known about ICH guidelines, whatever this total quality management from Q6 to Q12, all the quality guidelines are going to be involving with total quality management, right? So one part of the total quality management is our QbD, that is quality by design. That is, it is one part. That is, there are other things are also going to be there. That is, so this is what we are going to call as a quality. That is, so what is the quality of a pharmaceutical product? We are going, that is, I told you, the quality characteristics of a pharmaceutical product is, you see, the very important, the first and foremost one is, this is a therapeutic efficacy. So whenever if you are going to, it should give relief or disease, free from symptoms or from the disease to curing the disease or anything. That is. So that is what we are going to say, therapeutic efficacy one is. Another one is safety. Safety in the sense what I told you, minimum or no adverse effects. That is. So when the patient is going to be taking, maybe a little bit like a small adverse reactions that are going to be there, but it should be safe. That is, to take that one. And stability. These three are very, very important, because now all the pharmaceutical products, whatever we are going to manufacture, they are all manufacturing on a large scale process. Large scale process means we are going to manufacture the tablet not at 10 tablets or 15 tablets or 20 tablets, where we are having the old concept is dispensing concept, dispensing of pharmacy. That is, now it is totally vanishing, dispensing pharmacies. Now after getting the stability concept, now we are having a manufactured product. Previously we are going to call them as a dispensed product. Dispensed product means as and when you require, we are going to prepare the product in the retail pharmacy or whatever the pharmacy, dispensing pharmacy. This dispensing pharmacist will issue that dispense whatever that medication, whatever the medicine for 2 days or 3 days or 4 days. But now we are manufacturing the product, whatever the pharmaceutical product, on a large scale, and where the patient is going to be taking as and when he requires. That means it is going to be stayed in the retail pharmacy for a longer period of time under suitable storage conditions. That is. So once we are going to maintain the suitable storage conditions, we are maintaining its stability aspect. That is a long shelf life. That is, we are going to give expiry date now 2 years or 3 years. It depends on the type of the product. That is. So that is a shelf life is another important one. And from the industry point of view, that is pharmaceutical product quality, that is you, because from the patient should also get the product at a cheaper price, at a low cost. That is. So for that purpose, your technology, your excipients or whatever the process, all this be take it into consideration so that the patient will get benefited for that one, and it should be readily available. As and when he requires, he will go and purchase that one. Now in a corona pandemic situation. Now what is going to be happening? So many of the medicines are not at all available to the patient due to the lack of that supply of the medicines. So many patients are dying. That is very pitiable situation that is now we are facing in the pandemic situation. That is so readily available, that is the one important characteristic that is, and patient compliance, that is, yes, and then customer accessibility, that is another one, that is because at each and every corner of the globe it should be available, that is customer accessibility. He can purchase it from his native, from his village, that is, at least, that is nowadays we are having such a type of facilities are going to be available, that is. So these are all a quality of a pharmaceutical product, that is what we are going to. So this is going to be only possible when we are going to have, that is with a total quality management, total quality management. So in the total quality management, when we are going to discussing about this therapeutic efficacy, safety and shelf life, these three are very, very important for our today's discussion. What we are going to say as per ICH guidelines, what they have given, that is quality characteristics, that is by this quality by design. So what is the quality by design? We have already discussed, that is in the previous slide. So I will just go through quickly, that is the concept of what is quality by design, that is what we are going to have, that is, you see, quality by design is, it is what is our requirement. For this we require product design and process design, which we are going to discuss, that is. So you should know about the knowledge on the product design process, as well as process whatever the control, how we are going to get you, that is. Once you are having that product design and process design, means you are know about what are the process parameters and what are the quality attributes, that is of the product. That means desired quality of a product, we will discuss, that is right. So then if we are going to have a desired quality, that is you are going to specify, these are the product specifications, that is, and this is the process conformities. So once we are going to do all these things, then we are going to get a good quality product, right, with a product knowledge and a continuous improvement, that is, and process understanding also you should. So that is what we are going to say, that is quality by design. That is, for that design, what you have to do, that is, you see, designing of the product and its manufacturing process, designing the manufacturing process to consistently produce that product, because you very, very important, whatever the product we are going to producing, it should be consistently, we are going to, there is no question of any variation from batch to batch or from time to time or anything, that is. And you should have a very good sound knowledge on the understanding of what are the input parameters for the quality product, and what are the controls we have to maintain, what are the critical points we have to maintain in that process. That is, you should have a sound knowledge, what we are going to have, that is that sound knowledge, and whatever the process, whatever the understanding you should have, that is. So then we are going to have that whatever the product which we are going to design, it is a quality product which it can meet the specifications as per the requirements, that is. So such a scientific, logical, preemptive scheme is going to be nothing but your quality by design, that is. So this is what we are going to know about, that is quality by design. Now we understand, that is what is a quality by design concept, that is. So now coming to this quality by design, once we are having, that is, what are the elements present in this quality by design, especially with related to product development, that is, because this is one of the very, very important elements. That means how we are going to apply this quality by design principles, that is, because this is already, I think you already gone through all the previous topics, which we are going to, everybody is going to explain what are the principles, but here the principles we are going to apply to our product development. Which product? That is initially pharmaceutical product, then we will go for biopharmaceutical product. What are the how we are going to contemplate to the biopharmaceutical product, that is, right. So in that case, what are the elements in quality by design? I think you all know, that is, first the very important element is the QTPP, quality targeted product profile. That is one we have already known, quality target product profile. That is, from this quality target product profile, you are going to get what you are going to say, CQA, that is critical quality attributes. That means what are the important quality parameters which are going to be required for the three things we have to keep in mind, that is therapeutic efficacy, stability, as well as shelf life. That is, these three are very, very important for us. That is, so these critical quality attributes. So to reach that, whatever the critical quality attributes which are going to be showing its product quality profile, it depends on other two elements, what we are going to call it as whatever the raw materials we are using in the product, right? What are the raw materials we are using, that materials, including all type of materials, not only excipients, active pharmaceutical ingredients, including containers and closures, labels, everything that is, which we are going to, that all comes under materials. So among these materials, there are some type of materials which are showing, that means reflecting the quality of the product. If we alter that material, automatically the quality of the product is going to be altered. That is what we are going to call as a critical quality materials. That is materials, that is what we are going to say, that is right. So that is materials, what we are going to say. And then other one is process. When we are going to manufacture the process, doing the manufacturing the product, what are the parameters we have to control to manufacture that one, to get a quality product, that is process parameters. So four important things, that is critical, whatever the quality attributes, critical material attributes, and critical process parameters. So these are all majorly the four elements which we are going to come across. I will just show this diagrammatic representation. You see, the first one is what we are going to call as quality target product profile. That is, so what is your target? So you want to manufacture a biopharmaceutical product, or you want to manufacture a tablet which should have a conventional tablet. That means it should release the whatever the drug immediately. At what time? That is immediately within 30 minutes. That is your whatever the dissolution, that is. So such type of whatever you are going to say, that is quality target product profile. That is, so based on this quality target profile, we are going to say that your quality attributes. Quality attributes means if you want within 30 minutes, if you want a dissolution, means you whatever your dissolution time, that means T90, that is time required for 90% of dissolution. That is dissolution efficiency. In the same way, content uniformity, you should get a uniform content into the drug. These are all critical quality attributes, that is critical material attributes and critical process parameters. So these are all very, very important for this one, right? So if anything is going to be failing in its activity, that means if suppose the material is not meeting the requirements, that means it is fails. It fails means what? It shows its effect. That means if there is something wrong in the material, or if something wrong in the process parameters, so what type of failures we are going to observe? So what is the extent of that failure, right? How it is going to be affecting the quality of the product, that is. So these are all we are going to discuss. That is what we are going to call it as now. The new concept is in this one is risk. That is risk assessment. That is we are going to discuss in detail afterward. That is, so this risk assessment and design space and control strategy. Which means now the latest terminology is what we are going to call it as process analytical technology. And this design space and the risk assessment, these are all the three tools which we are going to apply. In quality by design. These are the tools. Tools means risk assessment, as well as whatever the design space what we are going to scale, and then control strategy, that is controlling process analytical technology. Afterwards we are going to discuss that is. So these three tools are going to be available in there. So for this, whatever the design space, yes, For the design space, actually the tool is, actually design space is not the tool. For the getting a design space, the tool is, the next one is what we are going to call it as design of experiments. Design of experiments is one tool. In the same way, risk assessment is another tool. In the same way, control strategy is another tool that is. So for this design of experiments is for fixing this design space. What is this design space? We will discuss afterwards, that is, especially with respect to biopharmaceutical product, that is, right? So now what we are having, that is a screening. This is design of experiments, that is whatever the variables we are using. That variables is, it may be materials, which are variable means which directly affecting the quality of the product. That is what we are going to call critical variables. Critical variables. That means if you change that one, automatically quality of the product is going to be altered. That is. So how we are going to identify whether that variable is going to be affecting the quality of the product or not? So that means all the variables must be first screened whether they are affecting the quality or not. Quality in the sense I told you, therapeutic efficacy, stability, and whatever the adverse effects are, whatever the safety precautions, that is, right? So these three are majorly important. Leave about the other one, that is, right? So now in this screening of these variables, whatever is design of experiments are going to be used, that is screening of design screening, that is, these are the experimental, because I am not going in depth about this design of experiments. We are going to screen the variables whether they are going to showing the effect on whatever the quality of the product or not by factorial, two-level factorial design experiments, or placket Burman design experiments, or two-level factorial design more than whatever the factors, that is less than 3 factors are this one. So first of all, you have to screen whether this factor is going to be affecting the quality of the product or not. Once you have screened, then yes, it is going to be affecting the product, quality of the product. Then what you have to do, how much quantity is going to be required. That is very, very important. So you are going to select a specific variable. That means a specific whatever the formulation ingredient which is going to be affecting, and what is the concentration, at what concentration it is going to give you the optimum whatever the quality. So that is what we are going to call it as design space. We have to fix it, their concentration levels, that is concentration levels. So this is going to be done by another type of, that is optimization process. That is, this is a screening design, this is optimization. So this design of experiments in the optimization, again we are going to use some factorial design experiments, 3-level, 2-level factorial design with center point, with center point f is equal to 3, or by this response surface methodology, or by this mixture design methods. So this is what we are going to design of experiments is a tool for fixing of your design space. So this design space is with respect to raw material, whatever the input materials, whatever the materials which we are going to use, that is critical material attributes, that is what I told you, that is, or you can also have the design space we are going to respect to process, that is in the next slides we are coming, that is how this process design we are going to design. But you have to keep it in mind, design space is going to be fixed by doing this tool, that is design of experiments, that is, right? So this is what we are having. And then this control strategy and risk assessment, these are the another two tools, that is which we are going to use in quality by design. That is, this is in the product development. We are going to once we are going to optimize the variables, then we are going to know about which factors are going to be causing the risk, right, means failures, so that we are going to prevent those failures, right? Or we are going to be reduce the failures so that the risk factor is going to be reduced. And then we are going to fix the design space so that this design space is going to be used in the development and manufacturing with a controls. With a controls means this is what we are going to call it as process analytical technology. Control strategy is going to be obtained. That is another tool. Process analytical technology is nothing but the use of rapid whatever the analytical techniques, rapid analytical technologies and control systems, that is use of rapid analytical technologies and whatever the process control and monitoring systems to control the product quality during manufacturing process. That is what we are going to call it as a process analytical technology, which we are going to come across this control strategy, that is right. So this is in just what we are going to say, that is a product development, that is in case of quality by design in product. But now what we are doing is we are going to develop our biopharmaceutical product. In biopharmaceutical product, what are the critical quality attributes? This is one important. What are the other critical material attributes? What are the critical process parameters? So these are all we are now contemplating in case of our biopharmaceutical product. So apart from this, what is our PAT, process analytical technology, that is, and what is the other one is design of excellence. How we are going to arrive this quality by design, that is what we are going to see in this case, that is biopharmaceutical development. What I told you, first you are going to have a biopharmaceutical, what is the CQA, critical quality attributes, you see, and design of excellence. So you have to first design space, you have to fix it by what are the critical quality attributes. I told you critical quality attributes means which are impacting the quality of the product, which attributes especially, and design of excellence, that is right. So these two are winning. So here the critical quality attributes in case of this one, the other one is PAT, process analytical technology. So in that, what I told you, process analytical technology, rapid analytical technologies, use of rapid analytical technologies, like what we are going to say, that is sensors, or using of online whatever the analytical techniques, that is for what purpose. And simultaneously these sensors are also going to be used to monitor the process and to control the process also, like especially we are going to discuss afterwards, so how the biopharmaceutical manufacturing product is going to be taking place. In that, we are going to use so many sensors and so many of the analytical techniques which are very rapid, so that what will happen, we can monitor and control the product quality in the manufacturing process, that is in the manufacturing process, so that we are going to get this quality by design. Once we know the quality by design, then we are going to give what are the control parameters we have to maintain, and what are the specifications we have to give. Specifications of your whatever the raw material or whatever the materials which we are going to use, as well as the other one is what are the process parameters, that is, you see. Now, once we have designed this quality by design, then we are going to get the specifications and process ranges, that is. Now, understand. So here, in case of the pharmaceutical biopharmaceutical development, the very important thing is the molecule. Because what are biopharmaceuticals, I told you, they are mostly proteinous in nature. Almost all biopharmaceuticals molecules are highly complex structures, that is, which are nothing but proteinous in nature. Monoclonal antibodies, or whatever the recombinant DNA products, which are nothing but proteinous structures, that is, which are maybe sometimes they are glycoproteins, or maybe lipoproteins, or maybe monoclonal antibodies. So all these are all not small molecules, because if they are small molecules, we can easily analyze, we can easily keep their whatever the monitoring and everything. But because of their complex structure and the activity of that whatever the molecule is very, very important. The activity of the molecule is mainly depends on its conformation structure, that is. So that's why the analysis we are going to not essay, it is going to call it as a potency of the molecule. So the potency, or what we are going to call it as a biological activity of the molecule, are going to be showing the therapeutic activity. So that therapeutic activity is not by doing just a simple chemical assay or anything. That is, if you are going for the biological activity, you have to determine. So for that purpose, you require a clinical experimental data. So once we are going to use that molecule into the existence, what is the clinical experimental data? That means once that is a molecule, whatever the manufacturing process, what is the therapeutic efficacy, that is, because this protein, some companies are going to be manufacturing obtained from a prokaryotic cell, and some companies they are obtained from a eukaryotic cell. It all depends on their whatever the manufacturing process. But what they are interested on therapeutic activity of the molecule. So the therapeutic activity of the molecule is mainly depends on a specific site where that activity is going to be taking place, not due to the entire weight of the molecule or anything that is, amount or anything that is. So for these protein molecules, the therapeutic activity is going to be determined, and the other quality is a preclinical data. Because why it is going to be happening is preclinical data means toxicological studies in the animal studies. So what type of molecule it is, what type of degradation of the molecule, we are going to come across that one also in the next slide. And then what about it, structure activity relationship, because the protein is a very, very important molecule, right? So because its therapeutic activity is going to be altered once its structure is getting altered. So there is a structure and activity relationship. Now the computational, whatever the drug design is going to be involved, especially in this one, what is this one? Structure activity relationship, computer aided drug design, that is. So when the structure is going to be all these, and once the whatever the protein structure, stability, that is. Stability in the sense that means the protein native conformatory structure is going to be altered due to temperature or due to pH or maybe due to some other whatever the materials which are going to be present along with this molecule, that is whatever your excipients or adjuvants which we are going to hear, that is. So all these are all what we are going to call critical quality attributes which we are going to take it into consideration. Based on that, we have to design of experiments. So that will give you the design space, that is. Design space means what we are going to say, that is, it should within that space we are going to perform our experiment, that whatever the manufacturing process or whatever the formulation development, it is going to be retaining the quality attributes, quality whatever the characteristics, that is. So for this purpose, during the process, if any change occurs, how we have to monitor? So for this purpose, process analytical technology, if any change is going to be taking place, that change is going to be immediately detected by online whatever the process technology, whatever the analytical technologies, rapid analytical technologies, I am using the word. So that means within minutes you are going to get what is the change is going to be taking place, rapid analytical technologies. And then if any process parameters are going to be altered, then immediately you are going to have some sensors. You see, nowadays we are going to have the sensors, NRI, near infrared spectro techniques are going to be used. Online process, whatever, when the mixing or in case of tablet manufacturing or anything, Raman spectrometry is going to be used for this online changes, if any there, rapidly, that is. So these process analytical technologies are going to be there for monitoring and controlling whatever the changes, and if any deviation is there, immediately we are going to coming to across this, whatever the, whether it is within the design space or out of the range, right? So that will give you, that is. So once we are going to have, then we are going to have, that is, yes, quality product by designing of. That's why what I told you, quality by design. Quality by design is a predetermined objectives. So you are going to be initially predetermined objectives. When you are having predetermined objectives, once you are having a sound knowledge on whatever your product knowledge and process confirmations, that is. Whatever that product knowledge as well as process, if you are having a sound knowledge, and then we are going to fix that whatever the design space, then automatically whatever your predefined objectives you are going to meet by keeping this process ranges and the specifications, that is. So this is what is going to be happening in case of biopharmaceutical development, that is very young. So for this biopharmaceutical, how it is going to be manufactured, because I am not going in detail about this biopharmaceutical manufacturing, because nowadays all these biopharmaceutical manufacturing of products are going to be consists of two steps, that is, right? One is by using this fermentation technology. In olden days we are going to call it as a fermentation technology. Now where we are going to use that fermenter, now we are going to change the name. Instead of fermenter, now it has been applied to, that is, a bioreactor. But means what we are going to do, we are taking that whatever the genetically modified organism, what I told you in case of in the first biopharmaceutical definition, in the by using recombinant DNA technology or by using hybridoma techniques, that is. So we are using this genetically modified organism or a genetically modified cell line. We are keeping in a vessel, and then we are providing nutrition to that whatever, because they are living cells. We are providing whatever the nutritive requirements for its growth and metabolic activity under controlled conditions in a large vessel. That is what we are going to do it in case of manufacturing process. That is, just I will show that one. What is the manufacturing process of a biopharmaceutical product? That is, you see, this is what we are going to, this figure I have obtained from the internet. I have not written the source, I forgot to give you, but this I have obtained from the internet. That is right. So this is what we are having, a bioreactor. So what I told you, a bioreactor is for growing of whatever the living cells. It may be a prokaryotic cell or it may be a eukaryotic cell. That is, so where we are going to, this prokaryotic cell or eukaryotic cell may be usually unicellular prokaryotic cell. That is, prokaryotic cells, bacteria are going to be used in case of recombinant DNA technology. Only in some cases they are going for Saccharomyces cerevisiae, that is a fungal organism is used, especially in hepatitis B vaccine. That is, but usually because E. coli organism is well established, its genetic characteristics are well established, so that's why mostly the host organism is E. coli, which is genetically modified. That is, so its gene sequence is going to be incorporated with a foreign gene, that is where the vector is going to be introduced into that one. That is, so that whatever it is going to be grown in a large vessel, right, by providing whatever the artificial, whatever the requirements. That means in vitro, what we are going to say, that is, we are growing outside the whatever the body, that is, that whatever the animal cell or whatever the microbial cell that we are growing in a large scale on a controlled conditions, right? So once we are going to provide all these nutritional requirements and whatever this one, then it is going to be producing a large quantity which can have the capability of producing that specific protein, right, whatever the recombinant DNA protein or maybe monoclonal antibody in case. So this is what we are going to call as a bioreactor. Once we are going to get, then the next step is, this is the entire is a downstream processing. Downstream processing means, I will just give you that very brief, that is, it is nothing but, that is the separation, identification, separation and purification of your desired product. That is what means. So any of whatever the fermentation process consists of mainly two steps. That is, one is upstream processing and the second one is downstream processing, right? So during this process there may be a chance of there are so many of related substances, product related substances, or process related impurities, or product related impurities. These 3 points you have to keep in mind. Afterwards we are going to discuss, that is, product related substances or product related impurities or process related impurities, which we have to take it into consideration in our quality by design product development as well as process development also. That's why I'm just giving. So these are all various unit operations, like what you are having, that is, this is cell lysis, cell disruption, right? And this is primary recovery, that means filtration process, or this is also precipitation and whatever the filtration process. That is, here ion exchange chromatographic techniques, that is, you are going to separate and purify your product, and finally you are going to get your by using a different chromatographic techniques. Now especially in biopharmaceuticals, the very important process unit operations are, that is, one is tangential flow filtration, that is one. Another one is size exclusion chromatography. Another one is ion exchange and gel permeation, that is, right, gel permeation chromatography. Various purposes are going to be there. I am not going in detail about that upstream and downstream processing. Now our aim is, once we are going to get our whatever the product, purified product, that purified product we are going to use it for our formulation and process, that is, right? So during the purification, what could be the chances of getting that impurities or related substances? For that purpose I am just giving this one, that is, okay. right? So once we are developed that, this is about manufacturing. Now coming to this whatever this formulation design, that means our main quality by design. Now you understand about what is a biopharmaceutical and what are the characteristics of a biopharmaceutical. Now we are now going in depth about the design space development in the formulation, that is. So design space development means, especially in the formulation, means with respect to materials, that is. Afterwards we will go for process. First we will go for that is design space for formulation. Formulation means with respect to API and with respect to whatever the qualities of whatever the API, that means active pharmaceutical ingredient, that is our whatever the proteinous molecules or anything. And then what are the risk assessment, that means what may be the failure of your active pharmaceutical ingredient, that means it may be degraded due to any parameters or due to the stability aspects or anything, that also we are going to discuss. And then finally we are going to fix your variables as well as other whatever the formulation excipients which we are going to come across, that is. So based on that we are going to, so the design space, how we are going to discuss, that is. You see, now I'll just target product profile I'm using. Now I'm just giving you one example in this one so that you will have it. Now you all know that now everybody is a biopharmaceutical, widely used biopharmaceutical. Can you just, anybody can, that is widely used biopharmaceutical nowadays in India? Huh? Which is I think may not be giving chance to our audience, which is insulin injection, that is. Now insulin is a biopharmaceutical where we are going to get a highly purified insulin in a large quantity. Nowadays it is going to be available, but in olden days it is not at all possible. It is partially purified, that is also animal insulin, that is. There are so many of antigenic reactions are going to be there, but now we are going to get a highly purified similar human insulin, that is by this recombinant DNA technology. So if you take that insulin injection, that is, so nowadays the insulin injection is going to be available in a pre-filled syringes. I think you all know about now the insulin syringes are going to be available with already some 100 units of insulin is going to be present in that one, that is. So how to develop, that means for that, because our aim is to develop such one, that means what is your target product profile? You require a stable insulin which we are going to store it at a 2 to 5 degree, 8 degrees centigrade, at least for 2 to maybe 18 to 24 months. Usually 24 months is the stability period there. That is what is your, and it is going to be what you are going to have, that is compatible with whatever that insulin, whatever the material, syringe material and the needles which we are going to apply that one, what type of needle and how the syringeability is going to be there. All these are all going to be target product profile, that is what we are going to say, that is we are predetermined our requisites, that is what we are going to say, product requirements and attributes. In that attributes we are going to say that insulin is going to be acting long acting or short acting or intermediate acting. That means insulin, plain insulin, that is clear solution, or maybe insulin zinc suspension, right? Like that we are going to say that is our product requirements. So that is what we are going to say, that is once you know about all these things, then coming to, you should have a sound knowledge on the product, that is molecule knowledge. Molecule means how the insulin molecule is, what is its characteristics, so how it's what is its critical properties, that means yes, it is storage, if what is its degradation. So if there is any change in the pH, it is going to be, there are some type of proteins, there are degradations which are going to be taking place in the molecule, deamidation or maybe oxidation, or maybe whatever the attachment of this sulfide bridges onto that one, that is there are so many of this one degradation are going to be there, that is possibility of this one, that is. And because so many of the not insulin protein molecules, they are having a post translational changes, that means glycosylation, that is glycosylation means what we are having, that is we are having a glycosidic linkage is going to be attached at FC molecule, FC region of the protein, that is whatever the monoclonal antibodies, that is. So like glycosylation or maybe oxidation or glycation, glucose molecule is going to be attached, that is. So these are all for various purposes, that is, they are going to be done in case of this protein. So you should have a clarity on that molecule knowledge, that is. Once you should have a molecule knowledge, then you are going to for what type of, because depends upon your target product profile, you are going to start your pre-formulation studies and choosing your excipients and everything. That also afterwards we are going to discuss, that is. And then if any type of degradation is going to be changing, so then you are knowing about what is the analytical assay and bioassay and whatever the pre-formulation studies and everything, that is. So these things are very, very important in the that's why product knowledge is very, very important. But this is how we are going to get this product knowledge. You should have a clinical data and preclinical data about its therapeutic efficacy, about its toxicity. Everything is going to be there because it is a biopharmaceutical. You have to keep it in mind, which we are going to obtain from a living tissue, that is, a living cell, that is, so that there should not be any type of impurities or any type of related products. If are there, how, whether they are going to showing any risk or not, that is going to be checked in case of risk assessment, that is. So in that risk assessment, molecular stability and formulation variables, that is. Molecular stability means the molecule must be stable, that is, right? So it should not be degraded, or the degradation is going to be there, and what type of parameters we have to maintain, that is. So first of all, this risk assessment is going to be done by one analysis. That is also one important tool in QbD. I think we are going to say that is in risk assessment, one tool that is which we are widely used method is FEMA, failure mode effects analysis, that is. What are the failure modes, that is, so which are going to be causing any type of failures due to this whatever the failures, in the sense which directly reflects on the quality of the product, that is, right? So this is what we are going to have risk assessment, that is what we are. So based on this molecular stability and formulation variables, then we are going to come to a formulation development, that is. So in that formulation development, once you fix the variables, once you fix that whatever the concentration and everything, then you are going for design of experiments. I told you, design of experiments is one of the very, very important tool for design space, that is. So then stabilization, that is, formulation stabilization is also. So how long it is going to be stable and everything. So this is what you are going for, this design of experiments and stabilization. Finally, you come to this design space. Design space means whatever the excipients, that is input variables which we are giving, those variables we are going to keep it in a certain concentration range with a specific type of specifications, so that it is giving a quality of the product. If it is deviating from that one, then the quality of the product is going to be deviated. So that is what we are going to put as a design space for in case of the formulation development. Yes, we are going to discuss in case of process development also the same design space also in case of process variables, that is, right? So that is what we are going to have, the formulation design space development. So here we are going to discuss about now coming to this risk assessment. I think this risk assessment is nothing but what might go wrong. We have to keep it in mind. What we are going to say, that is, and then if it is go wrong, right, what are the implications? So what are going to be taking place? Severity of once it go wrong, what is going to be happening? That means what about the quality of the product, right? And another important one is if it go wrong, it is going to be affecting the severity of the product. And what is the likelihood of getting this wrong? Likelihood of taking place, that means probability of this one, that is. So these three are going to be important in case of risk assessment, which we are going to come across in this risk assessment, what it is going to be there, that is. So you see, in the risk assessment, what is going wrong. So if it goes wrong, how it going to be impact on the quality of the product? That is impact. So how oftenly this wrong is going to be taken? That is what we are going to call as a likelihood. So this we are going to representing in another one. I will just say that is in case of a risk assessment also. I will just go through the next slide so that you will have a risk levels, right? You see. In case of categorizing the depending on likelihood and its impact, that is. right? Impact and likelihood, how it is going to be taking place. You see, some of them are whatever the acceptable risk. That means whatever is going to be happening in the future, that is what we are going to say that is. So broadly acceptable risk, no further risk is going to be happening. That is what we are going to say that is. So broadly acceptable risk, no further risk is going to be happening in the future. That is what we are going to say that is. So
Sorry for the inconvenience that has been caused due to the technical error. Our speaker will be joining shortly.
Hello.
Yes, sir. We can hear you now, sir.
Hello. Hello.
Hello, can we hear, sir? We can hear you,
Yeah,
sir.
yeah, yeah. From which slide? From which slide you got? I think I lost. How much time it actually? From the last 5 minutes or 10 minutes?
Yes, sir. Yes, sir. 5 minutes.
5 minutes, huh? So I
Yes, sir.
think I'll continue. That is from that slide. But
Yes.
you just tell me that slide, from which slide?
Yes, sir.
Hello. Hello.
Sir, it's
Hello.
from Formulation Design Space Development, sir.
Okay. Okay. Right. One.
Yeah, sorry. Risk levels?
Yeah. Yeah. That's that. This is over.
Yes, sir. Risk levels.
Yeah. I'll use. Yeah. This one completed. Huh? Hello.
Yes, sir. Yes. Risk.
Right.
We were in
Right.
risk levels, sir.
Okay. Risk levels. Huh? Next slide. Okay.
Yes.
Yeah. Risk
Yes,
assessment.
sir.
Right.
Yes.
Right. Right.
Yes.
So, right now it is audible, right? So sorry, I forgot. I
Yes,
have
sir.
not seen my mobile. I kept in silent mode. That's why I didn't. Anyhow, okay. Uh, so now, just I will go back to the previous slide where we are going to have, that is very important thing here. It is a risk assessment, that is. So in that risk assessment, we, especially in case of formulation development, what we are having is, what are the risk assessment, how we are going to take it, that is with respect to whatever the formulation that is. So in the formulation, when we are going to use a active pharmaceutical ingredient, that means whatever the product we are going to be manufacturing, that product is having an impurities or product related substances, that is. So these product related substances, what we are going to call it as, which are going to be impacting the quality when their concentration is going to be very high, that is. So that whatever that, because product purification, especially in the biopharmaceuticals, the product purification is a very critical step, and it is stringent precautions we are going to take, that is especially in case of whatever the protein substances. So in that, whatever the product related substances which are going to be left over, that one, they are going to be purified by so many of the chromatography techniques, but the level must be as low as possible, that is why, because such type of substances which are going to be directly impacting the quality, that means therapeutic efficacy of the product, then we are going to call it as a criticality factor, that is criticality determination, that is. So once at what level they are going to be giving that criticality determination, that is very important. So for that purpose, we have to monitor. Monitoring, routine monitoring is not required, but their percentage determination is going to be within the below level, that is very important, right? So that is a product related substances, that is. And whatever the product related impurities, that is impurities are again, they are continuously monitored, they are also criticality determination. So these are going to be take it into the risk assessment, especially in the risk assessment, what we are going to say, that is what might go wrong, that is very important. So which substance or which whatever this one is going to be affecting the quality of the product, that is what might go wrong, and what are the consequences if it goes wrong. That means how it is going, what might go wrong. Suppose something is going to be happening which is not affecting the quality of the product, you need not worry about that one. If it is go wrong, so that it is going to be affecting the whatever the quality of the product. The consequence is it is showing the quality degrading in the quality of the product. Then how oftenly it is going to be taking place, that is how often, that means what is the likelihood of going this one, is it going to be taking place very less number of times, or if it is going to be taking place very oftenly, or if it is taking place continuously, that is. So according to that we have to fix, that is the risk should be assessed properly, that is in the next slide we are going to discuss, that is this is with respect to product quality attributes, that is product quality attributes. Whereas when we are going to discuss about process quality attributes, that is impurities, that is pure process impurities, that is what is going to be happening during the manufacturing of the whatever this one, we are going to use a nutritional requirements from the host cell, for the host cells, that means for the microbes, or maybe to the whatever the cell lines, that is, or bioactive medium components, they are going to be affecting the quality of the product. Yes, definitely we are required, routine monitoring is required, that is impurities, that is. So these are going to be affecting the quality of the product, so that how it is going to be there, that is the severity, everything it is going to be noted down, that is. There are some impurities which we are going to use during the manufacturing process of fermentation process, medium components without biological activity, they need safety assessment. Hello, Sri Harsha. Hi. Hello. Is it audible? Okay. Hello. 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 Ratnamala. Hello.
Sir, it is audible, sir.
Audible. Okay. Huh? Right.
Yeah,
Yes.
yeah. It's clear, sir. It's audible.
Right.
Yeah.
Right. So the medium components without biological activity needs safety assessment. That means, you see, during the fermentation process, we are going to use sometimes during continuous stirring, during for 24 hours or 48 hours, foam is going to be developed. So for that purpose, in that whatever the bioreactor, we are going to add some type of anti-foaming agents. These anti-foaming agents are going to be left over maybe in the product, very small quantities, which won't have any significant effect on the whatever this one, but their whatever the impurity levels should not have any effect on therapeutic efficacy or the safety, that is. So their level should not be not more than this much, that is. When they are going to have, that is they are going to be given by one type of this one, impurity safety factor. Impurity safety factor is nothing but this is a non-biological components, that is non-bioactive components, that is. So that impurity safety factor is going to be determined by LD50 of that whatever the foaming anti-foaming agent or whatever the substance we are, lethal dose 50 divided by how much amount of impurity level which is going to be present in the product dose, that is. In that way we are going to calculate. For minimized safety risk is impurity safety factor should be always more than 1000, that is. So this is risk assessment, that is especially in case of this one. So these risk assessments are going to be taken into consideration for whatever our control strategy as well as for quality of the product, that is. That's why I told you risk assessment is nowadays it is a one important quality by design tool, that is. So this risk control is very important, that is right. So this is going to be differentiated by this one, risk levels. What now we have discussed, that is risk levels are categorizing depends on their whatever the probability of occurring, likelihood, and their effect on quality, impact, that is, you see, impact and likelihood. So that is high, medium and low, that is, and likelihood of going to be appearing. So if they are going to be at medium or low level impact and the likelihood is going to be there, but they are going to be broadly acceptable risk, that is, and no further investigation is needed, that is. If suppose that likelihood is going to be high with medium, that is whatever the likelihood is in the medium range, and high impact is going to be there, risk is acceptable, but investigation is needed to reduce that risk. That is very important, right? then sometimes the risk is unacceptable. Unacceptable in some conditions means they are going to be, their impact is very high and their likelihood is also going to be high. So then that risk is unacceptable and investigation is needed to reduce that risk. That is, so here the risk is acceptable, but investigation may be needed for that one. Here the risk is acceptable, but no further investigation is required. That is, so based on these likelihoods, whatever this one, risk management is going to be whatever this one. So this risk management is one of the very, very important tool which we are going to come across this one, right? So next, coming to whatever this formulation development, what we are going to take in place, especially in case of design space, what I told you, that is just what so far we have discussed, that is what I have just given you in this whatever this one. You see, in case of this one, you should have, once you are going to have a formulation, you have a prior knowledge and scientific insight about your whatever the target profile and whatever this your molecule knowledge, everything, clinical data and preclinical data and whatever the structure activity relationship, all these sound knowledge and scientific product knowledge should be there. Based on that, we are going to fix your whatever the excipient space by design space experiment, that is design of experiments. We are going to what type of experiment, what type of excipients you are going to use, and how, what are the concentrations and everything. So that excipients will have the impact on the stability, freezing, thawing behavior, because most of them are freeze dried products, maybe especially proteinous preparations, or maybe some of them are, you have to keep it in 2 to 8 degrees centigrade, some of them are in frozen condition. That is, so during, what about its freezing, thawing behavior, and how, what about its processability? Processability means some of them are lyophilizer powders. Lyophilizer powders, when we are going to prepare injection preparation, whatever this injections, so how, what about its solubility? So how it is going to be dissolved, everything, that is what type of solvents we are going to take. So this is all process also coming into one. So we have to take it into consideration of the structure of whatever that one, the structure of that whatever the bulk material which we are going to use, that is, and effective dose and lethal dose, and of the safety of the excipients also. That is what I told you just now, that is in the risk level management, what is the excipients effect. So based on this, we are going to fix the quality attributes. So whether it is going to be attaining the quality attributes with a good quality, then we are going for this formulation. That is right. This is the formulation aspect of this one. Now coming to this process, whatever the characteristics which we are going to take, that is process characteristics means once we are going to get that whatever the formulation variables and everything, we are going for compounding. Just simple, most of the whatever the biopharmaceuticals are either freeze dried products or maybe simple solutions, but compounding, that is. So in that, very, very important, the process variables are very, very important, which can adding addition of buffer or mixing of the whatever the solvents, right? So, or if it is a pre-filled syringe, or if it is, what about its syringe solution viscosity, or whatever the rheology, or how they are going to be filtration, because the very important thing in case of biopharmaceuticals is sterility and purity, because some of them are cannot be sterilized by terminal sterilization. So you have to go for aseptic filling. So during this aseptic filling, we are going to use for the filtration process. All these are all very, very important. So that we are going to take it into consideration. You see, drug product process, what I told you, that is the process, compounding process. So again, we are going for the risk assessment, FEMA, I told you. That is what we are going to say, the failure mode effective analysis, that is failure mode effective analysis. Based on that one, we are going to go for this design of experiments, So once we are going to have a process parameters by using this design of experiments, then we are going to have the design space definition. So what is this design space definition is, you will have a 3 regions. One is a characteristic region, another one is whatever your operating region, and whatever your optimum region is. So the characteristic region is where you are going to perform the experiments, and then you are going to fix your operating range, and then you are going to fix the set point, that is. So that is how it is going to be there. Product design space is going to be C. That is, this is the characterization range. So in up to this range, you are going to perform your experiments for how the risk and what is the quality of attributes and everything. So based on that, what you are going to fix it, that is, once you have done this characterization, this is all you are going done by this design of experiments, design of optimization process. So then you are going to go for this acceptable range. This is your acceptable range. Actually, your operating range is this one. So once you are going to do your whatever the process in the operating range, then it is perfect and it is going to give the quality attributes. Even though it is going to be going out of this operating range, but up to this range it is acceptable range. So that is the advantage with the design space, that is. So once your operation is going to be taking place in this range, nothing will happen. But if it is going beyond this, then your process has been changed, and again you have to process parameters, you have to change it, again you have to apply for regulatory whatever the acceptance, that is. So always the regulatory bodies will ask, your design space must be in the always that you have to confirm your acceptable range. Once you are going out of the acceptable range, then again you have to apply for whatever the regulatory whatever the license, that is, for changing of this whatever the design space, that is, right? So this is what we are having a process characterization range. That is how we are going to affecting this design space concept in case of process characterization, validation and monitoring. Just I am going to give you this is a process characterization. So initially you are going to fix all the process variables, anything what you are going to say. This is based on your process analytical technology as well as your requirements and everything. Then you are going to check whether the process is going as per the specifications or not. This is process validation. So once the process is going on, then your process analytical technology, what you are having, that is here, you are going to monitor all the variables are within the range and everything, that is. So once this cycle is going perfect, then once it is validation, then you are going to apply for regulatory requirements, regulatory filing for that QbD, that is, right? So this is what is going to be definition of the design space, establishing the quality control, whatever the control schemes, and then we are going to check, demonstrate the process can be formed at a commercial scale with the design space. Once it is going to be commercialized with the design space, you have to monitor for a continuously perfectly or not, monitoring the process performance. And then if any deviation is there outside the design space, and again you have to filing for extended design space, that is. If it is not there, it is maintaining in the process, then we are going for regulatory filing for your approval, that is filing of design space in terms of key and critical parameters. So what are the critical parameters and what are the design space, every acceptable ranges, all you are going to file to the regulatory body, that is. So this is what you are having in the case of design space concept, in case of process characterization, that is. So what are the process variables we are going to come across in this one? Just we'll go through this one. This is in case of process development, you are having a process knowledge and a scientific insight, that is, right? And then you are having a process parameters. So these two are very, very important, process parameters and material attributes during the process. What are they? What is the pressure? That means when we are going to be filling in the tanks from the syringe and to the filling whatever the bottle, pressure is very important. What at flow rate, what is, and what is the holding time, and what is the filter material, filter size, temperature and filling speed, that is. These are all process parameters. So these should be optimized. Again, design of experiments and the design space we are going to. For this, you should have a prior knowledge about such type of products are going to be there or not. Should have a knowledge and scientific background with respect to that whatever the molecule, that is. And material attributes, what I told you, what is the pH of that one, whether we are maintaining that online process controls, that is, pH is going to be perfectly, and whatever the viscosity, because in case of pre-filled syringes or maybe in any type of other solution products, whatever the rheological characteristics are viscosity, density and surface tension, and these are all going to be. Once we are going to have this, achieve this one, then how it is going to be impact on this machinability, because during the filling process, during the filtration, or during the filling, sorption process, or consistency, reproducibility, all these are all going to be impact on this one. This is the process parameter. Once this is going to be there, then yes, quality attributes we are going to getting, then product and process risk assessment, the same way which are going to be showing any type of failure modes are going to be there. So what is its impact on that one, whether it is showing a high impact or not. If it is showing high impact, we have to continuously monitor its whatever the controls and everything that is. So that is risk assessment in the process also very. Once we are having that one, then process performance or product quality attributes. So you are going to have your processes perfectly going on, and you are going to meet all your product attributes that is. So this is especially in case of a biopharmaceuticals, especially we are discussing about only formulation and process development only, that is right. Actually that whatever the material manufacturing is, that is entirely a large process, that is right. So this is what we are going to have in case of by using this quality by design. Nowadays we are going to have this is as per ICH guidelines, we are going to have that is quality by design Q5 to Q5A to Q5E. Exclusively they have mentioned that is biotechnological products quality guidelines, that is they have mentioned. Especially one important we have not mentioned here, viral clearance is one of the very important in case of this biopharmaceuticals, that is. So what are the advantages? I think you are already known about this one. Product and process, better understanding we are going to have, and we are minimizing the variations, effective technology transfer, and right first time and every time. That is one of the very important concept, that is especially from in the industry, that is you do first time rightly and then every time also, that is. This is with respect to product and process. From the industrial perspective point of view, it is timely launching you can product once you are going to have a quality by design in your process and product development. Continuous improvement is there, improve the yields and low cost and less investigations, because there is no question of any type of mistakes or anything, so that you need not go for investigations or anything. From the regulatory perspective point of view, reduction of post-approval submission, that is because once we are going to fix the design space, once we are going to say that it is in the operating space, then there is no question of going for reapproval for that one. Once you are going deviate that design space, then you have to go for whatever this one. And real-time release with regulatory, because the product is perfectly released in time through process analytical technology. Innovative process validation also we are going to say, and a greater regulatory confidence, that is regulator will have a more confidence about your robustness of the product based on this quality by design approach once we are going to submit for regulatory approval, that is. So these are the things. I think thank you very much for listening, patient listening. I think maybe a small hitch during this one due to the internet connectivity, that is right.
Thank you, sir, for giving a good insight into the topic.
Right. Hope I've taken a lot of time. I don't know.
And no, sir. We were it was very interesting to listen all these things. So we have some questions for you, sir.
Yeah. Welcome.
Sir, this is from Dr. Baya Subbarao Garu. Sir, for attribute, statistical confidence limits comes in operating or acceptable range.
Okay.
For an attribute, statistical confidence limits comes in operating or acceptable range. This is the question.
Please repeat. For an
attribute, statistical confidence limit comes in operating or acceptable range.
No, it should be in operating range. That is, first of all, that's why I told you, you have to fix your whatever the operating range first. You have to fix it during your design of experiments, and then you have to go for your whatever the acceptable range, that is. So always operating range should be your set point, right? And then it should be within this whatever the acceptable range. That is very important. Have you got my answer?
Yes, sir.
Right.
Thank you, sir.
Right.
Another question, sir.
Right.
On what basis do we select the design, mixture, factorial, or combined?
Yeah.
And is there any value for the likelihood to assess this risk?
Yeah. The likelihood risk is that is other one, but the design is, it depends on so many factors, that is. So I told you that is the design of experiments. It is a very vast one where we can go for, that is first the screening of this one is which variable is going to be affecting the quality attributes is, first of all, whether it is affecting the quality of the attribute or not, we have to check it. That is, whether it is going to be affecting, then we have to take it into consideration for its effect on the further optimization process. In that optimization process, you have to fix it. So whatever this, that's why I told you. So what might be go wrong, that is, you have to say, that is in the risk assessment. That is, if it is going to be definitely affecting the quality of the product, yes, we have to take it into consideration, and we have to see whether the risk is going to be acceptable or not. Right? Whether risk is acceptable, whether it is going to be routine monitoring is required, or any further investigation is required, that we have to take it. We cannot say that which design of experiments we have to go for, means it all depends on a lot of exercise, that is lot of trial and error experiments in the formulation and development. That is, so by optimization of whatever the thing is, you have to go for depends on what are the number of variables which are going to be affecting the quality of the product. Depends on that which design of experiments we are going to go for, that one, that we will decide. That is, number of variables of very high, actually the design of experiments are going to be, lot of interactions are going to be there. In such conditions, it is not possible for all at a time. That is, you have to go for at least in a factorial design experiments, or fractional factorial design experiments, or whatever that surface methodology which we are going for this whatever the box Benken design, or maybe another one is that composite design, that may be, or this may be. It depends on the number of variables. That is right.
Thank you, sir. Another question,
Right.
sir.
Right.
Sir, please kindly touch upon analytical quality by design, and how is it different from ICHQ2?
Analytical quality design, I think it is different from actually in biopharmaceuticals, the analytical quality design is entirely different. Especially, I'm just talking about with respect to biopharmaceuticals. In biopharmaceuticals, this is, we are mainly interested on not the assay, that is assay in the sense that is, we are not taking the whatever the weight of the substance are in, we are taking the therapeutic activity of the molecule. That's why I told you, a protein molecule, not the entire molecule won't have any effect on that one. It is on the specific site where it is going to be attacking to that whatever the receptor, where it is showing its biological activity. So here we are analytical assay in case of this biopharmaceuticals is biological assays, that is mostly, or immunological assays we are going for, that is antigen antibody reactions, right? Whatever the process are going to be there. So that is separate analytical techniques in case of whatever the quality biopharmaceuticals quality, that is there. That is there. That's why I told you it is in case of Q5 A2 Q5E, that is what they are going to be in viral clearance. Another one is in case of Q5B, I think so. I not exactly same, that I cannot say. Expression vector analysis. What is that expression vector analysis means? Actually, the gene which is responsible for producing that protein is very, very important. So that gene sequence, everything they are going to be tested. Once that gene sequence is perfect, then that protein is going to be produced perfectly. That is, once that protein characteristics are there, then its therapeutic activity is perfect. That is, so that is Q2 is a little bit different from this biological activity. That is right.
Yes. Sir, another question, sir.
Hello.
Does QbD completely replace quality control?
Yeah.
Does
Ratnamala,
QbD
breaking. I am
completely
getting breaking. QbD
does completely replace quality control?
No. Not completely. Quality control is compulsory. We have to do it. That is end product, finished product testing. We have to submit that results. That is, it is not replacing anyone. It is now the quality control is one small part of whatever the entire total quality management. That is, in that entire total quality management, QbD is in one part. It is not a quality control is replacing the QbD is replacing the quality control, but there is must quality control is for regulatory requirements. We have to, because we are giving a label claim. On what basis we are giving label claim? Finished product testing only. Okay.
Yes, sir.
Hello.
Sir, another question.
Right.
Sir, another question, sir.
Yeah.
In case of live or attenuated vaccines, how to remove the contaminated virus from the product?
This is a very large process. It all depends on how the virus is getting entered into our product. That is the main question. That is first one. So how the virus is going to be enter into our product in a case of live or attenuated vaccines, that is, it is means whatever this live or attenuated vaccine, bacterial vaccines we are going to talk about. If it is viral vaccines, means other virus contamination, that is. So here the virus contamination is mainly due to your whatever the ingredients I told you, host cell, whatever this one is going to be prone to this virus contamination, or sometimes that whatever the components which we are going to use for the growth of the cells, especially serum. Serum is the most prone to viral contamination. Calf fetal serum or anything, that is. Right. So these viral clearance is now actually, as per ICH guidelines, that is what we are going to say, that is 5A. That is the first one is ICH guidelines. Q5A is viral clearance. Virus can be one way inactivated, or one way it can be removed. Removed means either it can go for by some type of ion exchange chromatography or by filtration. Now the filtration is also going to be processed, that is. So the viral removal is either inactivation or removal. It all depends on the type of the product, what type of product, and what is your manufacturing process, and all that is that we are going to take it into consideration. That's why this is one of the very important risk assessment in case of process, that is. In the process, that is, I told you, that is in the process, whatever the risk variable is, this viral contamination. Okay.
Yes, sir. Thank you, sir.
Right.
So thank you, sir. Thank you for sparing your time in this busy schedule and giving us good insight into this topic of application of QbD to biotechnological products, formulation and development. On behalf of RBVRR Women's College of Pharmacy, I take opportunity to thank Professor Girja Shankar Garu for giving a good talk on this topic. Thank you everyone for joining us on the live. Thank you, sir. Thank you, sir.
Right. Thank you very much for giving me this opportunity, and thank you for all the audience for patient listening. That is okay. I hope I have give a small piece, that is because I am not expertise in biopharmaceutical manufacturing or anything in the research or anything. With my small knowledge in that one, I just what I learned, that I have given to you, that is right. So thank you very much for giving me this opportunity, for RBVR College management and the principal, Dr. Sumakant, and the other organizing committee, especially I forgot the name, chairman Shailaja.
Shailaja. Yes, sir. Yes, sir.
Krishna Shailaja, and her team. That is, thank you very much. That is right. Thank you.
Thank you, sir. Thank you.
Right. Okay. I'm leaving.
Yes.
Hello.
Yes, sir.